Hello class, so we come to the third lecture on metabolism. So the Krebs cycle or citric acid cycle or the tricarboxylic acid cycle is here. So we have glucose and fatty acids, amino acids and all those things. They can be channeled through the citric acid cycle, starting by producing acetyl coenzyme A. And the process whereby it produces acetyl coenzyme A or acetyl coenzyme A is what we call the pyruvate dehydrogenase complex. So it passes through the pyruvate dehydrogenase complex. Pyruvate dehydrogenase. Pyruvate dehydrogenase complex, and this is a complex of enzymes, a multi-system enzymes, different enzymes, about 5 of them. Hydrolipoid, something hydrolipoid, whatever. There are about 5 enzymes, and they convert each of the one pyruvate to acetyl CoA. Acetyl coenzyme A, acetyl CoA or acetyl coenzyme A, acetyl coenzyme A. So this acetyl coenzyme A is what enters the Krebs cycle, and it joins with oxaloacetate. Acetyl coenzyme A will join or react with oxaloacetate, and oxaloacetate is a 4-carbon molecule or compound. So an acetyl coenzyme A is 2 carbon. We all know that acetal is 2 carbons. Acetal is 2 carbons. That is CH3, CH2. This is acetal. Acetal. If it is like this, it is acetate. If it is like this, it is what? Acetic acid. So 2 carbons. So we are considering the acetal group, CH3, CH2. So instead of we having acetal group being joined to 2 oxygens, making the acetate, we rather have what? Coenzyme A. That's CoA, acetyl CoA. So this is what we have. So this will be channeled. The acetyl CoA will be channeled. This acetyl CoA, this acetyl CoA will be channeled. This acetyl CoA will be channeled through the Krebs cycle to go and react with what? A 4-carbon compound, oxaloacetate, to produce 6-carbon compound, citrate. Citrate will remove water from citrate to produce what? Isocitrate. When we eliminate water from citrate using the enzyme called aconitase, then we'll have isocitrate. Then we further reduce isocitrate by removing 2 protons or 2 hydrogens from it to produce what? Alpha ketoglutarate with the release of carbon dioxide. So we have, under the auspices of the enzyme isocitrate dehydrogenase, alpha ketoglutarate will add coenzyme A to alpha ketoglutarate, and we further oxidize or remove protons, 2 protons from alpha ketoglutarate, and add them to nicotinamide adenine dinucleotide, add them to produce reduced nicotinamide adenine dinucleotide. And it is catalyzed, this reaction is catalyzed by alpha ketoglutarate dehydrogenase. So this enzyme will move two hydrogens and add them to oxidized nicotinamide adenine dinucleotide to produce succinyl coenzyme A, because we added coenzyme A here, and we remove CO2 or carbon dioxide. Succinyl coenzyme A synthesis. Anytime you see synthesis, an enzyme being a synthesis, it means that it is able to synthesize or produce a product. And synthesis is able to synthesize or produce a product with the expenditure of what? ATP. So a synthesis, synthase and synthesis. Synthesis. Synthesis uses ATP to synthesize a product, but this one does not use ATP. Does not use ATP. So there are two enzymes. The synthase uses ATP to synthesize or produce a product or catalyze a reaction, but synthesis uses ATP. Synthase does not use ATP. So mark the difference. Note the difference. So here, succinyl coenzyme A, when we remove this coenzyme A, the succinyl coenzyme A synthesis will catalyze, will move out of coenzyme A from succinyl coenzyme A to produce succinate. And here we produce GTP. And GTP is a high energy compound like ATP. So the GTP here is equivalent to 1 ATP molecule. Then succinate is further oxidized by the removal of two hydrogens or two protons to produce fumarate. So we can see the coenzyme FAD removes two protons from succinate, catalyzed by succinic dehydrogenase. So that we have FADH2, reduced flavin adenine dinucleotide. Then fumarate, when we remove water from fumarate, we produce malate. Then when malate is also oxidized by removal of two protons, we produce NADH under malate dehydrogenase. So whenever you see a dehydrogenase enzyme, any enzyme having dehydrogenase means that it catalyzes oxidation reduction reaction. Either, most of the time, it removes two hydrogens, removes two protons, from a substrate, adds two protons to a substrate. So this is what we have. So in the end, we produce back oxaloacetate. So let's now look at chemiosmosis or oxidative phosphorylation. Oxidative phosphorylation. Let's look at oxidative phosphorylation. Well, this is a process whereby it's also called the electron transport chain, or chemiosmosis. So let's see what it entails.
Professor Dave again. Let's take a look at oxidative phosphorylation.
He knows a lot about the science stuff. Professor Dave explains.
acid cycle. As it turns out, neither process generates much of an energy payoff, but the NADH and FADH2 that were generated in the citric acid cycle move on to oxidative phosphorylation, which generates by far the most ATP out of these pathways. This pathway utilizes an electron transport chain, which is a series of mitochondrial membrane proteins that sit in the inner membrane of the mitochondrion, and we refer to these as protein complexes 1 through 4. These proteins bear a variety of prosthetic groups, which are non-protein components that give the protein its functionality, including flavin mononucleotides and cytochromes. There is also one compound in the electron transport chain that is not a protein, and that's ubiquinone, a small hydrophobic molecule that is mobile within the membrane, and it is also known as coenzyme Q, or CoQ. Once NADH feeds electrons into the first component of complex 1, these proteins facilitate a series of redox reactions, shuttling the electrons downhill from one component to another, with each structure down the chain having a higher affinity for electrons than the last. This process does not generate ATP directly, but a byproduct of this series of electron transfers is the generation of a proton gradient across the membrane. Protons accumulate outside of the inner mitochondrial membrane, which then go to power another protein complex called ATP synthase. As you might guess from the name, this is the component that synthesizes ATP. Because the proton concentration becomes greater in the intermembrane space than inside the mitochondrial matrix, the protons will spontaneously move with the gradient to re-enter the mitochondrial matrix, and the only route available is through ATP synthase. This process is called chemiosmosis, and because the gradient is able to do work through chemiosmosis, we also call this the proton motive force. And it is these protons that power ATP synthase in phosphorylating ADP to generate ATP. ATP synthase has a fascinating structure with a component that looks startlingly like a rotor, where individual protons can bind and cause it to spin in such a way that catalyzes phosphorylation of ADP, kind of like a stream of water turning a water wheel, which can then power some other process. With the NADH and FADH2 that is generated from a single molecule of glucose, we can get around 26 or 28 ATPs from this activity. So this is the pathway that generates the majority of cellular energy. So in summary, your body uses a variety of metabolic pathways. Glycolysis, the most ancient, produces 2 ATPs per glucose, but it also results in pyruvate, which can enter the citric acid cycle. Here, those 2 pyruvates generate another 2 ATP, but they also generate 6 NADH and 2 FADH2 molecules that can go on to the electron transport chain, which gives us our big ATP payout. Feel free to use this summary of the 3 steps in cellular respiration if you need to remember the main points of each process, all starting with just 1 molecule of glucose. Other sources of food, like proteins, fats, and other carbohydrates besides glucose, are initially broken down in unique ways, but this will always result in some compound that is eventually fed into one of the pathways we discussed. So all the food we eat eventually gets broken down this way. This is why mitochondria are regarded as the engine of the cell, since most of the energy needed for cellular activity comes from them. So the next time you're walking home and you feel tired, just think of all those trillions of enzymes slaving away to help you move your feet. It might put a little pep in your step. Thanks for watching, guys. Subscribe to my channel for more tutorials.
So, the precursors of oxidative phosphorylation, which is also the same as electron transport chain or chemiosmosis. We have glucose being transformed to pyruvate to produce acetyl-CoA. It can also be fatty acids, amino acids, and the rest being transformed to what? Acetyl-CoA. Pyruvate can go through the oxidative phosphorylation, or amino acids and the rest can all go through, fat or fatty acid, they can all go through oxidative phosphorylation. Before that, they have to go to the pyruvate dehydrogenase complex to produce acetyl-CoA, which will now react to produce reduce nicotinamide adenine dinucleotide phosphate and FAD, which will now go into or be channeled into the oxidative phosphorylation pathway or electron transport chain. So, a summary of oxidative phosphorylation or electron transport chain. So we have these are complex 1, complex 2, complex 3, complex 4. Complex 1, 3, and 4 are proteins. They are mitochondrial membrane proteins. Matrix, the membrane protein. Then, ubiquinone is not a protein. Ubiquinone is not a protein. And it's also called coenzyme Q. Coenzyme Q. Ubiquinone is called coenzyme. It's a coenzyme. Coenzyme Q. You remember your cofactors and coenzymes. This is ubiquinone, also called ubiquinone. It is what? Ubiquinone. Ubiquinone is a ketone. It's a ketone. It's a ketone. Ubiquinone. Okay, so ubiquinone is coenzyme Q. That is here, complex 2. So, like you saw in the video, this proton will come and there will be electron transfer from complex 1 to complex 2, complex 3, complex 4, cytochrome C to complex 4. Then after passing through complex 4, we will have what? Protons will go out over here. Then the electrons will pass through the complex, the 4 complexes. So when the electron passes through the 4 complexes, we will have, in the end, we'll have 2 electrons here. That's for one cycle. Or we have 2 electrons here, then we have 2 protons here, because these protons will also, when they pass here, they will come through the process. Over here we have the chemiosmosis over here. The protons will be pumped. After they go through these complexes, they will come and they will travel down this big complex. And this one is catalyzed by ATP synthase, will produce ATP. So this is ADP with 2 phosphate groups attached. Then this is ATP with 3 phosphate groups attached. So the protons will come down their electrochemical gradient through this complex. And this one is what we call the chemiosmosis. So when it comes like that, the final acceptor of electrons, the final electron acceptor is molecular oxygen. Here we are seeing only 1 oxygen, but they are supposed to be 2. So that's why we have times 2. Molecular oxygen is the final electron acceptor. So let's see. In the electron transport chain, when they ask you that, what is the final electron acceptor? It's molecular oxygen. So it will accept 2 electrons. 2 electrons plus half oxygen, half O2. Another 2 electrons plus half O2. Half O2 is the same as what? Oxygen. 1 oxygen. This is also the same as 1 oxygen. So the final electron acceptor is what? Molecular oxygen. So the molecular oxygen will accept 2 electrons. So we get what? O2. O2. So sorry, we will get here 1 oxygen here. 1 oxygen here. Then we'll get another oxygen also here. Then we have protons are 2. 2 protons here, 2 protons here. So in the end we are producing 2 molecules of water. 2 molecules of water. So we started from, so here H will be 2. 1 proton here, 1 proton here, another proton here, because the electrons are 2, so the protons are supposed to be 2. So 2 protons, 2 protons, 2 electrons, 2 electrons. So this one will be H2. These 2 protons will produce this H2. Then these 2 protons will also produce this H2. Then, so when we add these 2 electrons to these protons, these 2 electrons also will add to these 2 protons to produce H2. And this H2 will add to half O2, which is O, to produce what? Water. And the water that will be produced here is one water molecule for each side. So this one will produce 1 water molecule. And this one will also produce what? 1 water molecule. So in the end, we'll have what? 2 molecules of water being produced. So 2H2O. Let's go. So in the end, we have what? 2H2O. ADP is converted to ATP. We added phosphates to ADP, making ATP. When the protons are going through this complex, this chemiosmotic complex, we produce ATP. And more energy is produced in oxidative phosphorylation or electron transport chain than glycolysis and also the citric acid cycle. So that's oxidative phosphorylation taking place in the intermembrane space, inner mitochondrial space, and also the matrix. So let's see. So these are the complexes: complex 1, complex 2, complex 3, complex 4, ATP synthase. So the last one is ATP synthase. That's the enzyme here. The enzyme here is called ATP synthase. This ATP synthase does not use ATP. It produces ATP, but it doesn't use ATP to produce ATP. If it were a synthesis, then it will use ATP to produce ATP. So the same reaction. So over here we have the electron transport chain, because we are transporting electron through complex 1 to coenzyme Q, which is complex 2, to complex 3, cytochrome C, complex 4. Then before the protons will pass through the chemiosmotic channel under the auspices of ATP synthase to produce more ATP. And this one occurs in the mitochondrial matrix. So the same steps. So we are seeing, I've already explained. So the protons will be 10. Let's see the mechanisms. So let's see. Okay, so we have 10 protons. So the protons are 10 here. 10 protons here. And we use ATP to produce more ATP. So the things that can inhibit okay, so these are some things that can inhibit this electron transport chain: retinol, actinomycin, cyanide complex, oligomycin. They can inhibit it, so they cause uncoupling of the enzymes or the protein complexes. So energy yield, we have glycolysis. Glycolysis will produce 2 ATP. 2 ATPs. Pyruvate metabolism will produce what? 2 NADH. 2 NADH. So times the 2 NADH, the energy in the 2 NADH is 2.5. So times 1 NADH will produce 2.5 energy. So times 2 will give us 5. Pyruvate metabolism, 5. So for pyruvate we have, we produce 4 of this, so we have this. TCA cycle will produce 2 ATP, so the energy is 2. TCA cycle also uses 6 NADH, and each NADH has what? 2.5 energy. So 2.5 times 6 is 15. 2 FADH2 1 FAD produces 1.5 energy, so times 2 gives us 3. So the total energy from glycolysis to the TCA cycle and even to the electron transport chain will be 32 ATPs. ATP, this is a trial question, go and solve it. Okay, so thank you very much. Let's move to class for our research presentation. Thank you.